In this lecture, we're going to look at the concept of capillary exchange from chapter 21. Basically, we're going to look at three processes. One of them, obviously, much more in depth than the others, but the first and most important means of capillary exchange is just simple diffusion. That's just materials moving from areas of higher to lower concentration. You'll see a variety of different components found within blood move or be exchanged via simple diffusion. Items such as oxygen, carbon dioxide, glucose, various amino acids, and the list can go on. But you should definitely keep in mind, like I said, the first and most important means of capillary exchange, although we don't even spend much time discussing it, is going to be simple diffusion. The main reason, obviously, we don't discuss it at length is because you probably, or you should have already seen that concept, obviously, during general biology. The second means of capillary exchange I want to mention here is called transcytosis. With transcytosis, it's basically two processes that you've been exposed to before, but those two processes are coupled together. They are the process of endocytosis, in which materials are taken up by a cell, and then exocytosis, in which those materials are released. So basically, transcytosis translates out to transverse or go across a cell. So by endocytosing whatever material is being moved and then exocytosing it, you are basically allowing that material to move across or through a cell. You see this a lot with the placenta and placental transfer of various nutrients from mom to baby's bloodstream. Now, the final aspect of capillary exchange that we're going to talk about is going to be called bulk flow. Bulk flow, although it's not the most important means of capillary exchange, is very important when we start to evaluate fluid balance. When we look at capillaries, they exchange fluid with the surrounding fluid, the fluid that surrounds them, which is called interstitial fluid. And based upon whether the fluid is a part of interstitial fluid or a part of your blood as a part of plasma, it can tremendously affect how things work within the human body. Now, just to give you an illustration, when you have too much interstitial fluid, for instance, that causes swelling, or what we technically call edema. Obviously, swelling isn't a good thing, or edema isn't a good thing. So, this process of bulk flow helps in part determine where the fluid is. Is it a part of the interstitial fluid, or is it a part of plasma? And if it's not occurring correctly, there can obviously be negative consequences, one of which, an example of, is edema. So we want to talk about bulk flow and what all plays a role in determining that. Now, when we look at bulk flow, once again, it's the movement of fluid or an exchange of fluid between the plasma and also the interstitial fluid. Now, the interstitial fluid, if you don't remember, is fluid that surrounds your cells within your tissues. So we're looking at fluid moving back and forth. I want to emphasize that point because when we see fluids move back and forth, they do so, well, at least according to physics, due to pressure differences. So that fluid will always move from an area of higher to lower pressure. The pressure that drives the direction of movement of the fluid, we refer to it as net filtration pressure, or NFP for short. And if that NFP, net filtration pressure, dictates that fluid leave the capillary and become a part of interstitial fluid, we refer to that process as filtration. Once again, filtration is the process via which fluid leaves plasma or leaves blood and becomes a part of interstitial fluid. Now, the dynamics can also change. And should the net filtration pressure dictate that fluid instead goes from the interstitial fluid into the blood or into plasma, we refer to that process as reabsorption. So essentially, I've tried to define for you what the process of filtration and reabsorption are. Filtration and reabsorption are the two arms or the two components to bulk flow. Filtration is the situation in which fluid leaves blood or leaves plasma and is going to enter the interstitial fluid. Then reabsorption is the process of taking interstitial fluid and allowing it to re-enter the blood. Now, if you take a look at this image, you can kind of see when that fluid is filtered out, it would take with it the nutrients to supply those tissues, to supply those cells. And then when it's reabsorbed back, it should bring with it waste products. Now, there are several forces playing a role in determining net filtration pressure. 4 total forces overall, 2 of which are generated by the blood, and 2 of which are generated by the interstitial fluid. If we look at blood, the first pressure we consider is called blood hydrostatic pressure, BHP for short. The hydrostatic pressure of a fluid is the fluid itself exerting a pressure. And the hydrostatic pressure will always try to cause the fluid to go away from where it currently is. So we consider blood hydrostatic pressure. This is once again a pressure determined by blood. And since the hydrostatic pressure tries to make the fluid go away from where it is, we see the direction of the arrow. BHP tries to make blood leave the capillary and enter interstitial fluid. The other force arising from blood is called the blood colloid osmotic pressure, BCOP for short. We call it blood colloid osmotic pressure to specifically relate to only one of the solutes found within blood. You know, there's many different solutes, obviously. There's glucose, there's various electrolytes or ions, there's amino acids, and water-soluble vitamins. All this stuff can be found dissolved in blood or dissolved in plasma. But we don't really concern ourselves with those other items. The one that we're going to principally concern ourselves with during this process of bulk flow, plasma proteins. And the colloid in blood colloid osmotic pressure tells us specifically that this pressure is related to plasma proteins. Now, there can be two reasons why fluids will move. One could be the fluids themselves trying to move. That's a hydrostatic pressure. But in the process of osmosis, we see that the solutes within a solution can also try and dictate where fluid will move. And that's exactly what happens with the osmotic pressure. The solutes within that particular solution will try to pull fluid to where they are. They try to make it become a part of the fluid that the solutes are in. So blood colloid osmotic pressure, it's the plasma proteins trying to influence the interstitial fluid to become a part of blood, to pull that fluid from interstitial fluid back into blood. And notice the direction of the arrow here. So those are your two forces associated with blood. We have a blood hydrostatic pressure trying to drive fluid out of the vessel, and a blood colloid osmotic pressure determined by the plasma proteins trying to pull fluid in. We still have an osmotic and a hydrostatic pressure associated with the interstitial fluid. They're very negligible though. Once again, we see interstitial fluid osmotic pressure. This would be due to the solutes in the interstitial fluid, and they're going to try and pull fluid from blood to where they are. Now, we don't have to only concern ourselves with with proteins in the interstitial fluid. There really shouldn't be many proteins in interstitial fluid. And the reason is because plasma proteins don't really get filtered via a capillary. If you recall the structure of a capillary discussion, I told you one of the items that's limited when we start to look at the exchange of nutrients and waste across a continuous and even a fenestrated capillary, one of the things that doesn't get filtered is going to be plasma protein. So we don't really see many proteins in that interstitial fluid. The interstitial fluid hydrostatic pressure is usually very close to zero. You can see the hydrostatic pressure, remember I told you a few moments ago, tries to make the fluid go away from where it currently is. So since we would be talking about interstitial fluid, we see the direction of the arrow here moving into the vessel. What we do in order to calculate net filtration pressure is summarize all the forces driving filtration and subtract all the forces driving reabsorption. So blood hydrostatic pressure and interstitial fluid osmotic pressure both drive filtration. Blood colloid osmotic pressure and interstitial fluid hydrostatic pressure both promote reabsorption. So this becomes our equation for net filtration pressure. Blood hydrostatic pressure plus interstitial fluid osmotic pressure minus blood colloid osmotic pressure plus interstitial fluid hydrostatic pressure. Now, at the arterial end of a capillary, what happens is the pressures will be slightly different than they are at the venous end. So let's consider the arterial end first. At the arterial end, our blood hydrostatic pressure will be roughly 35 millimeters of mercury. Our interstitial fluid osmotic pressure is 1. And we subtract from that 26 plus 0 because blood colloid osmotic pressure is 26 and our interstitial fluid hydrostatic pressure is 0. And we'll get plus 10 millimeters of mercury. This positive 10 millimeters of mercury is the force driving filtration at this arterial end. But if you look at it, there's one number out of this set of 4 numbers that will be greater than the others. It's the 35. And I usually say this, or make the statement that the high man on the totem pole wins. In other words, the highest pressure is the dominant pressure. It is what drives that actual process. So one concept that I definitely want you to keep in mind is this. When we look at the arterial end, blood hydrostatic pressure is the highest out of all four pressures. Therefore, it is blood hydrostatic pressure that technically drives filtration. As BHP goes, as blood hydrostatic pressure goes, so does filtration. Now, that blood hydrostatic pressure, although it is linked to your blood, is due to one significant parameter within your body. That's your blood pressure. So, when we consider the process of filtration, what is it? It is the process of fluid leaving blood and becoming a part of interstitial fluid. It is driven by your blood hydrostatic pressure, and that blood hydrostatic pressure is determined by your blood pressure. That's kind of the thought process that you should be going through when you consider this process of filtration. Let's go ahead and look at the opposite end of this capillary and start to talk about the process of reabsorption. As we move through the capillary and filtration occurs, you lose more and more fluid. As you lose some of the fluid contained within blood, the blood hydrostatic pressure goes down. Remember, blood hydrostatic pressure is a force exerted by your blood, and if you don't have as much blood, which happens if you lose some of the fluid, then your BHP will drop. So what was 35 at the arterial end becomes 16 at the venous end. Remember, I told you a few moments ago, high man on the totem pole wins. Well, now if we look at all four numbers on this venous end, the blood hydrostatic pressure, being 16, is no longer the highest number. It has now become 26 that's the highest number, and that 26 corresponds to our blood colloid osmotic pressure. We do the math and we can see that we get, instead of a positive number, a negative number here. Negative 9 millimeters of mercury. And whenever NFP is negative, that indicates reabsorption. Once again, high man on the totem pole wins, so it is the blood colloid osmotic pressure that drives reabsorption. And keep in mind, I told you earlier, blood colloid osmotic pressure is determined by plasma proteins. So essentially, it's the plasma proteins that drive this process of reabsorption. So to summarize kind of some things with reabsorption, similar to what we just did with filtration, reabsorption is the process of fluid coming from the interstitial fluid back into blood. It's driven by blood colloid osmotic pressure, and that blood colloid osmotic pressure is determined by plasma proteins. Now if you look at it, this positive 10 and negative 9, these numbers are not in balance. There's a little bit of extra force pushing fluid out, and then you have reabsorbing the fluid back in. This means that there's an imbalance in the amount of fluid filtered versus what you reabsorb. On average, you filter 20 liters of fluid per day. You only reabsorb 17 due to that imbalance. So 3 liters of fluid is left behind. That extra interstitial fluid, the 3 liters that's left behind, is accounted for by your lymphatic system. And that's all the talk that we'll have in chapter 22 when we look at the lymphatic system. But you should understand that, as I talked to you earlier, this condition called edema is a negative situation. And usually edema is caused by one of two major things: either too much filtration or inadequate reabsorption. So when we start to look at edema, for instance, if your blood pressure goes higher, one of the things that may potentially happen is that you may swell or have edema. Just think about the last time you ate crawfish. When we usually eat crawfish, the sodium within the boiling mixture causes our blood pressure to increase, and you'll usually get swelling of the hands and the feet. That additional pressure, the increase in blood pressure, increases blood hydrostatic pressure, and our net filtration pressure, instead of being 10, may go to 11 or 12, forcing more fluid out, more fluid than we can reabsorb, and we get edema as a result. Another probable cause of edema could be inadequate reabsorption. There are many diseases that can affect the function of the liver, which is where many plasma proteins are made. If the liver does not work, then it doesn't make plasma proteins. And consequently, as that plasma protein level decreases, we may see edema. We could consider the situation where a person may have liver failure. One of the things that usually develops during liver failure is swelling of the hands and the feet, along with other areas of the body. And that swelling is all a result not of too much filtration, but of actually inadequate reabsorption, because the liver is no longer making the correct amount of plasma proteins. So that's the discussion here about capillary exchange. Please keep in mind those 3 major types, and obviously you should keep in mind the points that we made about the process of bulk flow.